Hello, my name is Zara Assar. I'm the lead scientist of structural biology services at Cayman Chemical. Today I'll be presenting our work on a recombinant SARS-CoV-2 neutralizing antibody which blocks the Spike RBD-ACE2 interaction via biophysical characterization using SPR. SARS-CoV-2 is an enveloped positive stranded RNA virus, a member of the beta coronavirus genus, and a causative agent of COVID-19. As of September 2021, the number of COVID-19 cases worldwide had reached over 223 million. In addition, the number of deaths from COVID-19 was over 4.3 million worldwide. Among vaccines development, neutralizing antibodies against the SARS-CoV-2 spike glycoprotein receptor binding domain have been explored. Spike glycoprotein is located on the outer envelope of the virion and is composed of two subunits, S1 and S2. This one subunit contains the RBD, which binds to the angiotensin-converting enzyme 2-ACE2, followed by S1 and S2 subunits cleavage by TMPRSS2 to facilitate viral fusion with the host cell membrane, followed by infection. Envelope protein, membrane glycoprotein, nucleocapsid protein, RNA, and spike glycoproteins are structural components of SARS-CoV-2. S1 RBD domain of spike glycoprotein binds to the ACE2 receptor. Therefore, human neutralizing antibodies have been identified that bind to the S1 RBD, preventing further viral entry and infection through disruption of the S1 RBD-ACE2 interaction. However, this virus has been rapidly mutating and now has several variants labeled as variants of concern, according to the World Health Organization, WHO, and Center for Disease Control, CDC. In this study, variants effects on ACE2 binding were analyzed, specifically N501Y, K417N, and E484K S1 RBD mutations effects on a recombinant SARS-CoV-2 neutralizing antibody were studied. First, we made recombinant human antibody expressed in CHO cells using engineered constructs based on sequences of antibodies from COVID-19 patients. Of one of the most frequently elicited antibody families, immunoglobulin G heavy chain variable gene IgHV 353 isoforms CC12.3 and CC12.1. The binding of this recombinant antibody to the S1 RBD and its ability to block the S1 RBD-ACE2 interaction were characterized using surface plasmon resonance SPR. Our goal was to evaluate the direct binding kinetics K on and K off and KD to optimize target engagement on rate and residence time on a target off rate directly. Also, to establish a SPR platform to evaluate binding kinetics and affinity measurements for the neutralizing antibody against S1 RBDs and its ability to block ACE2 interaction. And lastly, to establish competition assay between SARS-CoV-2 neutralizing antibody, Fab region, and ACE2 against S1 RBD to determine whether the neutralizing antibody has reduced neutralizing capabilities towards these variants versus wild type. We needed to optimize SPR protocol for S1 RBD wild type, alpha and beta variants with SARS-CoV-2 neutralizing antibody, Fab region, and ACE2, and running single cycle kinetics assay for ACE2 to S1 RBD, single cycle kinetic assay for SARS-CoV-2 neutralizing antibody, Fab region to S1 RBD, running single cycle kinetic competition assay for SARS-CoV-2 neutralizing antibody and ACE2 to S1 RBD, and lastly, multi-cycle kinetic competition assay for SARS-CoV-2 neutralizing antibody, Fab region, and ACE2 to S1 RBD. Based on SPR results of single-cycle kinetic runs, ACE2 had a similar affinity towards S1 RBD wild type, alpha and beta variants, with a KD of around 9 nanomolar and a slight difference in association and dissociation rates for beta variant. SARS-CoV-2 neutralizing antibody, Fab region, had a very high affinity towards S1 RBD wild type, alpha and beta variants with a KD of below 1 nanomolar and a slightly better affinity towards wild type. And lastly, for the competition assay, SARS-CoV-2 neutralizing antibody, Fab region, could provide better blocking against ACE2 for wild type and alpha and beta variants respectively. We then followed with a multi-cycle kinetic run for the competition assay to look at neutralizing antibody Fab's region ability to neutralize S1 RBD wild type and variants when ACE2 is added at KD, 4 times the KD, and 1/4 of the KD concentration. We are showing data for ACE2 at 10 nanomolar concentration, or the KD concentration here. Overall, the data correlated well with the single cycle kinetic run, and neutralizing antibody Fab region could provide better blocking against ACE2 binding for wild type and alpha variant with IC50 of 64 nanomolar and 68 nanomolar respectively, compared to the beta variant for IC50 of 120 nanomolar. The same results were obtained using in vitro and cell-based assays. Essentially, this engineered recombinant antibody can neutralize wild type better than alpha and beta respectively against ACE2 binding. Lastly, we wanted to know how this recombinant neutralizing antibody can bind to the SARS-CoV-2 spike glycoprotein and block the ACE2 interaction. Therefore, we tried CryoEM study in collaboration with Thermo Fisher Scientific. We were successful in obtaining the CryoEM structure of the SARS-CoV-2 spike glycoprotein bound with Fab region of this neutralizing antibody at 2.9 angstrom. CryoEM study was performed by Eva Drew Light at the Thermo Fisher Scientific. Interestingly, based on this cryo-EM structure, Fab region of the SARS-CoV-2 neutralizing antibody could bind to the open conformation of the S1 RBD of each chains. Basically, knowledge of these structural data will have implications for the next generation vaccines and antibody therapies towards the new variants of the virus. In this study here, we wanted to investigate SARS-CoV-2 neutralizing recombinant antibody blocking the S1 RBD-ACE2 interaction using biophysical technique SPR. We made recombinant neutralizing antibody, human ACE2 receptor, and SARS-CoV-2 S1 RBD wild type invariants in-house using our established mammalian expression system. Further characterized their binding via surface plasmon resonance SPR using our BioCore instrument. We could establish a SPR platform to evaluate binding kinetics and affinity measurements for ACE2 and recombinant neutralizing antibody towards SARS-CoV-2 S1 RBD. Lastly, we could successfully determine cryo-EM structure of the SARS-CoV-2 spike glycoprotein in complex with the neutralizing recombinant antibody Fab region in a collaboration with Thermo Fisher Scientific. Thank you for your listening.